I'm very proud to announce the first interviewee in this series of interviews. He is somebody who is right at the forefront of MND research and makes decisions on where the money that you guys help us raise is spent. And it's really an honour to be able to speak to him today and ask him some questions about MND research. So, without further ado, I'll say hello there to Dr. Nick Cole. Hi, Nick.
Hi there. How are you doing today?
I'm good, thank you. I'm very good. Could you just start us off by telling us about your role at the MNDA and what a typical day looks like for you?
Yeah, absolutely. So my position title is Head of Research, and I am in the Research and Development Directorate, and that's headed by Brian Dickey, who's the director of the team, really. My role is to look at research communications, making sure that we sort of understand and have an oversight of all the research that's going on around the country and around the world, and to try and distill that science down and put it across in lay terms so that people can really understand what we're doing as an association in the fight against MND. In addition, I'm also involved in organising the International Symposium, which is one of the largest, or the largest, MND specific conferences in the world. Last year we had 1,800 delegates, and the idea of that is to bring research scientists and healthcare professionals together to make sure that we get that information and those new discoveries across as quickly as possible, and that information gets shared and nobody's working in silos, and to just facilitate that collaboration and those across the world projects, really. And also to have an oversight of our grant applications, and I take part in our BRAP panel, which is our biological research applications process, which is looking at the grant applications, and they're all peer reviewed internationally by experts in the field, to see where the funding should go to try and find those discoveries.
Our videos get viewed by people who some people who've had a lot of experience and the knowledge about MND, but equally people who are kind of may just have been diagnosed, may have been looking at symptoms, may not have been diagnosed, or have just heard about a family or friend or loved one. So we're going to start off quite basic, if we can, please. I'm just going to ask you, what is motor neuron disease?
Yeah, so motor neuron disease is a condition which can affect anybody. It doesn't discriminate, and it's a disease of the motor neurons, hence the name motor neuron disease. And motor neurons are really the electrical wires that connect from our brain to our muscles. So when we want to do a voluntary muscle contraction, for example, moving your little finger, you'll send a message from your brain all the way down these neurons or electrical wires into the muscle. And that electrical signal allows the muscles to contract, because the nerves anchor into the muscle. And it's the degeneration or loss of those motor neurons or electrical wires, as it were, that causes the disease. And because muscle is a tissue that we can change with exercise, for example, weightlifting, you can make the muscles bigger or change the types of fibers in a muscle, depending whether you do weightlifting or run a marathon. Conversely, if the muscle isn't used anymore, it actually starts to atrophy or waste away. So, for example, if your arm's in plaster, or you were fortunate to go up to space and spend 6 months on the space shuttle, space station, because you've got that lower gravity, the muscle isn't used as much and it wastes away. So what happens in motor neuron disease is, because the motor neurons are not allowing that muscle to contract and the muscle's not being used, it starts to waste away. And so, unfortunately, those voluntary muscles include muscles of the diaphragm, which affect breathing, the voice and swallowing, and also movement of the arms and legs. So as we're all very aware, it's a really devastating condition. In addition to the fact that it's called motor neuron disease and it's a disease of the motor neurons, there is substantial evidence now that there are lots of other cells around the motor neurons that maintain them and make sure that they stay healthy throughout our lives. So lots of work now going into looking at those other cell types that exist to nourish the motor neurons and make sure that they remain healthy. They are very specialized cells. They can be up to 1 meter long for a single cell, which is an incredible length for a single cell to be. And there's a lot of energy and maintenance that's required to make sure that those long cells stay healthy and stay alive. And also, in addition, we can't actually regenerate motor neurons. So a lot of our other cell types can be regenerated. For example, if you cut your finger, it grows back you might get a scar or you may not. But that regeneration doesn't happen in motor neurons. So if something starts to affect them and they start to die or degenerate, they never get replaced. So that's the thing that we're trying to understand in terms of motor neuron disease, looking for effective therapies.
Absolutely fascinating. Thank you for that. Do we know what triggers the disease, and if particular people are more susceptible to it?
In some cases, there are families where several members of the family have had motor neuron disease, and that indicates that there is some sort of genetic link potentially in the disease. And we know in around about 10% of cases of motor neuron disease, there is a familial link, or it is passed through the family, and there's a known genetic cause. So currently, the only thing we actually know causes motor neuron disease or is associated with causing motor neuron disease is a few particular genes and changes in genes which people with the disease have and people without disease don't have. There have been a multitude of other theories and searches for other factors. We know that aging obviously is something that increases your risk of motor neuron disease. The older you are, the more likely you are to get it. But also there are a whole plethora of theories and ideas ranging from things like amalgam in fillings, living under electric wires, gut biome, diet, blue-green algae. There's many things have been looked at, but really nothing's actually been tied down as an actual cause of the disease.
It's one of those things that's always people ask me kind of, how did I get it kind of thing, and it's an impossible question to answer really, because I don't know.
I should just say that just because you have one of these gene changes as well doesn't actually mean to say that you'll develop symptoms. So that's another layer of complication. So there are other factors which may be protective or mean that you don't actually trigger those symptoms. So yeah, it's a multifactorial state where we're trying to understand all of those different things.
Give us an oversight about what's currently available to treat the disease.
Yeah, so unfortunately, there's very little available in terms of effective treatments. There are only a couple of compounds. Only one is licensed in the UK, which is riluzole, which we know is effective and does extend life for a period of time. But again, it's far from what we really need in terms of an effective treatment. So there's a desperate need to find effective therapies.
Could you tell us how research projects are generally set up and then funded?
Sure. Yeah. So we have a call for applications initially, and so researchers will submit a summary application. So, for example, they would have some preliminary data and an idea and a concept, and they would like some money to do this research, to employ the right people to do the job, to get the ingredients they need and the equipment, for example, to make that project work. So they'll submit a set of summary application, and we may get 30 of those or 40 of those summary applications from all around the country, and some internationally as well if they're linked to some UK institutions as a collaboration. And those summary reports get sent off to experts in the field, and they come back with an opinion as to whether this is a potentially a really good idea, or an idea that needs a little bit more work, or should be rejected as not a suitable project to be done. And on the back of that, we then invite a larger application. So those people that pass that first phase will then put in a long application, and then that gets sent out to international experts all around the world again, and gets peer reviewed, and it gets effectively marked on many different criteria. You know, the feasibility of it, whether it's based on good preliminary data, whether somebody else has been doing it somewhere else in the world, so it wouldn't make sense to do the same thing again. And then those projects all come together with a score, and we have our scientific advisory BRAP panel, which all sit together, and one person is a spokesman for each of those projects, and they'll explain to the rest of the group, who will have also read the grant, why it's been scored a certain way, and some of the pitfalls and some of the positives of the project. And then there's a discussion goes around the table to really sort of deep dive into that particular project. And then at the end of that process, it'll get scored, and all of the projects will get ranked. And then, unfortunately, we only have the funding out of, say, 30 applications, we would really only have the funding to fund, say, 4 of those projects. So it's a lot of work for people to put in those projects, and a lot of rejection. And we'd give feedback as well, so people can improve on the projects they put in. But ultimately, that international peer review process means that only the really suitable and sensible, good projects get funded in the end. So it's a rigorous process, and one that takes a lot of time, but it's worth it to make sure the best projects get funded.
What are the top 3 challenges in researching MND?
Great question. Yeah. So I'd say, first of all, MND, I believe, is not incurable. It's just underfunded. So one of the major challenges is to get enough funding to do all of the research that's needed to answer all of the questions. I think that's really, really one of the key points. And then probably as part of that as well is to sort of retain the best and brightest people as well. I mean, one of the things about doing research is you're always applying for money and funding to keep doing the work you want to do. And you can train somebody and have 3 years funding, and then if that money runs out, that person will then go off to work somewhere else in another area or another disease, and so you've lost that expertise. And so it takes a bit of time to catch up, I think. So that's another area, but again, that's all down to funding. And then I would say, the barriers really would be the potential complexity of the disease. It's been worked on for a long time, and we're really making some great discoveries and really pushing at the door, but it's not a simple disease. There will be an answer out there, and it may be a really simple answer in the end, but people are working really hard to find out what that answer is.
How much does it typically cost to fund an MND research project? And how important is charitable funding for that project?
Yeah, so to fund, well, how long's a piece of string? So, I mean, some of our small grants, can make a real difference. So in terms of funding, say the supply of some samples that are flown across the world to be analysed, the repercussions from that data can be very far-reaching and last a huge amount of time. So that could just be a few £1,000s to do that. Most of our full research projects would be for 3 years, around about the £250,000 mark. So that would be 3 years. Normally, that would be funding the salary of a couple of lead researchers, maybe a PhD student, providing all of the ingredients and components they need to do those experiments, funding to run the microscopes. You normally have to pay per hour to use these incredible microscopes, and computer time. If there's animal models involved, you'd be paying for the animal house costs. You've got to feed the fish, for example, or grow the cells in dishes and feed them with media and things like that. So that level is really set by ourselves. And if we could have £1 million grant, then we would potentially offer that, and then the scientists would need to justify why they need £1 million for that grant. But normally So a 3-year grant would be about £250,000. PhD studentship, which would be just funding the scholarship of the student, some of the funds would be a little bit less. So yeah, it's an expensive business, and I guess we're quite a big employer in that respect.
Our first donation for research purposes went towards a project which was called MND's Next Top Model, evaluating iPSC models according to protein aggregation pathology. Now, can you tell us, in as simple terms as possible, what that actually means?
Yeah, absolutely. So, I mean, one of the really key things about developing therapies is that we need to really understand the biology of the disease. So it's very difficult to treat something if you don't really understand what the cause is. And to study motor neuron disease in a person is very, very difficult, because obviously we can't see the motor neurons in a person and see what's going on. So we need to do what's known as modeling the disease. And that can be done using an animal model, such as a fish, a fly, a mouse, or in a dish. So it's now possible, due to some incredibly clever people in the past, who have worked out a way in which we can actually grow human motor neurons in a dish. So this is really sort of almost science fiction turned into reality. So it's now possible to take a skin cell from a person's arm, and to put those cells, after you've broken up that tissue, put those into a dish, add a few factors, and you turn some of those cells into what's known as pluripotent stem cells. So it's a stem cell, meaning it's got the potential to make any tissue. So you're almost winding the clock back on those cells. So you've got these iPSCs, which are induced pluripotent stem cells. That's the iPSC part. And then it's even more incredible that you can then add these 4 different compounds, factors, to the fluid that the cells are in, and they'll actually go down a motor neuron disease pathway in development. So those cells will become motor neurons. So we're actually able to make human motor neurons in a dish. And this has gone even more advanced now, in that we've got human motor neurons, but we're now also able to put those other cells around the neurons, and also muscle cells even on the other side of some very small, what are called microfluidic chambers with very tiny holes, and those neurons actually will grow in and join the muscle. So you've effectively got a 3-dimensional model of nerves and muscle in a dish. That's all very great, but in terms of motor neuron disease, taking that to another level higher, is that you could take skin cells from a person with motor neuron disease, turn them into neurons which will effectively have motor neuron disease, and compare them to skin cells from a person who doesn't have motor neuron disease. So you've got that genetic background in a person, from a person with motor neuron disease in a dish, versus the control. Now there's another layer on top of that, which you may have heard of CRISPR-Cas9 and these new gene editing technologies, which allow you to make a change in some of the errors in a particular gene. So what we can now do with these iPSC cells is you can take the skin biopsy from a person with motor neuron disease, grow their motor neurons in a dish, but also using those same cells, you can use this CRISPR gene editing technology to make what are known as isogenic controls. So you can correct the genetic mistake in the cells from the person with motor neuron disease. So and then grow those into neurons in another dish. So you've effectively got 2 dishes. One is the person with motor neuron disease neurons with the genetic error, and in this dish you've got the same motor neurons grown from the person with MND, but you've corrected the genetic error. And then you can do lots and lots of different experiments on the 2 dishes, the same experiments on each dish, work out what the differences are in the biology and in all the things that go on, and also test compounds and therapies to see whether there are differences. And there are some one straightforward example is you can label fluorescently, so you can see it down a microscope, stress proteins, and you can see that in the cells with the genetic error on the person with MND, there is a much higher level of these stress proteins and these aggregates and sticky clumps of protein within the neurons than there are in the control cells, or effectively normal motor neurons. And then you can look at that to analyze potentially how compounds might dial down that stress protein and that response. So yeah, it's kind of Star Trek type amazingness, but being used in a really powerful way for MND.
Absolutely fascinating. I mean, it's truly incredible what you're now able to do. I mean, the developments in technology that have allowed you to do those sorts of experiments, it must have accelerated and just changed the scope of what you're able to do in MND research compared to even 10 years ago, for example, I would imagine.
Yeah, that's right. It's a huge advance in technology, and this is happening all the time. You know, we're taking all that information and skill from other areas of science and other areas of technology. You know, I mentioned earlier about genes and sequencing and computer power. You know, the ability to process massive amounts of data is really important now in science, and it's based on a lot of these genetic principles. And without those advances in technology, you know, we'd never be able to do that work.
Absolutely fantastic.